10th CBSE Biology, Chapter Reproduction. In this video, you will learn vegetative propagation and tissue culture. Roses, the most beautiful among flowers, and also the gardener's favorite. Rose plants always find its place reserved in all gardens. But unlike other flowering plants, roses are not grown by seeds. Instead, stem cuttings. It is same for sugarcane and also hibiscus. This special method of reproduction is called vegetative propagation. Vegetative propagation is a kind of asexual reproduction. It can be defined as asexual method of reproduction in which a new organism develops from vegetative parts of the organism or a parent. So the vegetative parts of the plants give rise to a new plant. That is what is happening here. Stems are the vegetative parts of the plant. Vegetative parts are the parts of the organism which are non-reproductive in nature. It means that they do not take part in reproduction. So those parts of the body of the organism are called as vegetative parts. Now if we consider a plant, the vegetative parts of a plant include shoot tip, node, leaf, stem and roots. These are all the vegetative parts of the plant. Vegetative part means the non-reproductive part or the parts which do not take part in reproduction. Let us see more examples. Here are two more methods of vegetative propagation, grafting and reproduction by stem modification. Grafting is a horticultural technique in which the parts from two different plants are joined together so that they grow as a single plant. Here you can see the example of tomato on the screen. Observe here, these are potatoes here and tomatoes in this region. A single plant bears both tomatoes and potatoes. Now how is it possible? It's possible because of the technique called grafting. In this technique, what you are seeing here is the shoot of the tomato is grafted to the root of a potato plant. The new plant has a shoot which gives tomatoes and the root which produces the potatoes. So a single plant has both tomato and potatoes. Hence the name pomato. This is one method of vegetative propagation. The second method what you are seeing is the stem modification. The runners and the stolons send out the modified stems and these produce new plants. Here you can see this is the new plant and down you can see that there are roots. Here you can see there's one more plant which is growing. So these modified stems which are called as runners produce the new plants as they grow. The storage parts of the plants also produce a new plant, like for example ginger, onion or potatoes. Bryophyllum is one more example of vegetative propagation in which the vegetative buds in the margin of the leaf produce the new plants. When these buds fall to the ground, they produce roots and start growing as a new plant. Now you see the growth of plants in an artificial medium. This technique is called tissue culture. Tissue culture is an artificial or induced vegetative propagation. Tissue culture is carried out in a laboratory. It's a technique of growing plants from cells and tissues of a parent plant in an artificial medium. Here you can see this is the artificial medium, or it can also be called as nutrient medium. It is called artificial because it's made by man in the laboratories, and this contains all the essential growth factors or nutrients required for the growth of plant. Now here you can see in this slide the stages of tissue culture. The first step is to select a part for tissue culture. Here in this case the leaf is selected and a tissue sample is taken from the leaf and is placed in an artificial medium. Artificial medium. This artificial medium has growth factors which will lead for the development of the tissue into a callus. Callus is an undifferentiated mass of cells. When we tell undifferentiated mass of cells, it simply means that the cells are young and energetic. These young and energetic cells are capable of multiple divisions and hence they go on multiplying and lead to the formation of a tissue or a cell mass. This undifferentiated mass of cells is called callus. And from this the cells are isolated and each cell develops into a new plant. Here you can see in this test tube there is a plant which is grown. Please observe, in the beginning it was a leaf tissue, then it got transformed into a callus, then the cells were isolated from it and a new plant developed from the isolated and cultured cell. Note that it was from a tissue sample which was taken from the plant, a new plant has developed. Hence, this method is called as tissue culture. Tissue culture is one of the asexual methods of reproduction, but this time induced by man. Once the plants develop in the test tube, they are later transferred to the nurseries and from there to the fields. Observe the image carefully. It's a single tree with multiple fruits. The tree is called fruit salad tree. This is now a reality. Yes, you heard it right. Fruit salad tree is now a reality. This has been possible only because of the techniques of grafting, tissue culture and vegetative propagation. Dig in deeper to know more about the amazing applications of these techniques in agricultural sciences. In this video, you learned vegetative propagation and tissue culture techniques.